Hello to all chicken lovers and future poultry genetics experts. If you're already familiar with our content on our wonderful Ornamental Birds channel, you know I'm passionate about sharing valuable and useful knowledge on various specific crosses for producing high egg-laying hens. Today I'm especially excited because we're taking the first step on an incredible journey, a Basic Poultry Genetics Course. Yes, you heard that right. We're going to demystify chicken genetics, from the most basic principles to how those highly efficient hybrid egg-laying hens we see today are created. And the best part is that you'll acquire this valuable knowledge completely free, right here on our YouTube channel. Forget about paying for expensive courses on other platforms. Here, quality poultry breeding education is at your fingertips, with the same passion and expertise that has always characterized us. If you've ever wondered why a chick hatches a certain color, how the most productive laying hens are obtained, or how to predict the colors of your own offspring, then this course is for you. In this first chapter, we're going to lay the groundwork, unraveling the secrets of chicken plumage color. Get ready to discover the following. Where do all those colors come from? How are the basic color palettes of our chickens formed? And how a few master genes can change everything when performing certain selective crosses. So without further ado, let's get comfortable and begin our genetic journey. We're going to start by talking about the two base pigments red and black. These are pheomelanin and eumelanin. Imagine you have a painter's palette, but with only two basic colors. In chickens, all that incredible diversity of plumage, from jet black to the brightest gold and pure white, is built with only two types of paint or pigments, which are the following: pheomelanin. Think of this pigment as the warm red on the palette. It's responsible for all the red, golden, orange, yellow, and soft cream tones. If you see a hen with intense mahogany plumage, that's the work of pheomelanin. A clear example would be a Rhode Island Red rooster or hen, especially from the exhibition line, which has a dark mahogany color. Eumelanin. This is the intense black on the palette. It gives rise to black, elegant gray, and interesting blue colors. When a hen is completely black, it's because her feathers are packed with eumelanin. Both pigments are created by tiny color factories called melanocytes, which deposit these pigments directly into developing feathers. The magic isn't just in the colors themselves, but in how they're used. The intensity of these colors can vary enormously. For example, a red can be a vibrant mahogany or an almost white cream, and a black can be glossy or a very pale gray. All of this depends on how much pigment is produced and how it's distributed along and within each feather. And a curious fact, if a chicken, for genetic reasons, doesn't produce either of these two pigments, its plumage will be pure white. Sometimes this is due to albinism, or a condition called leucism, which we'll explore later. What's interesting is that even if the plumage is white, the color of the legs and beak may or may not be pigmented independently. Genetics are amazing. The key to understanding colors is knowing that the presence, absence, intensity, and, above all, the distribution of these pigments are controlled by multiple genes that work together. Now let's look at the basic color palettes of chickens, the magic of black distribution. If pigments are our paints, then chickens come with a predetermined basic color palette in their genetics, which dictates how these paints will be used on each feather. Here, the main artist is a fundamental gene we call E, for extension of eumelanin or black pigment. The different versions of this gene tell the chicken exactly where and in what quantity black pigment will be deposited on each feather. Any areas not painted black will be filled in by red or gold pigment, which is pheomelanin. Think of eumelanin black as the ink that the E gene distributes on a canvas, with the feather being that canvas. Depending on how it extends or restricts it, we'll get a different palette or basic pattern. Let's look at the solid black color palette, EE. Here, the E gene commands the black pigment to extend almost all over the body, covering most of the feathers. The result is predominantly black, shiny plumage. Any red or gold pigment the hen has is suppressed or very limited, appearing only as slight reflections. Some lines of Australorp or Minorca are classic examples of this color base, being completely black. Imagine a canvas almost entirely painted black, where the black ink has spread widely. Then we have the palette called the Birchen palette. In this palette, the E gene still extends a lot of black pigment, but strategically restricts it in certain areas, allowing red or gold pigment to express itself in a very contrasting way. You'll see a mostly black body, but with a touch of gold or silver if there are other dilution genes, on the breast, hackle, neck feathers, and/or wings. The dark Brahma is a great example of this pattern, where the contrast between the dark body and the silver or gold markings on the neck and breast is very distinctive. A black canvas, but the black ink has left a clean, defined space for brushstrokes of gold or silver. The Wheaton palette. Here, the E gene further restricts the extension of black, especially on the body. In the rooster, the body will be primarily black, but with a very pronounced cape, hackle and saddle feathers of intense red or gold. The hen, on the other hand, will show a light brown or wheaten body due to pheomelanin with a salmon or reddish breast. Tail feathers are usually dark. The Rhode Island Red has this wheaten base, although it has been selected for generations to extend the red and make it more solid and uniform. Also, the New Hampshire, similar to the Rhode Island Red but with a lighter red tending towards buff, and many barnyard chickens have this coloration. Imagine a canvas where the black ink is more restricted to certain areas, leaving ample space for warm tones of red or wheat. The wild partridge palette. This is the most ancestral palette, the original pattern of the wild jungle fowl. The E gene here is the least restrictive in terms of eumelanin on the general body, but it generates a specific drawing pattern on each feather. The rooster is very striking, with a dark body and very bright red and gold hackle and saddle. The hen exhibits an intricate pattern of lines and layers on each feather, known as partridge lacing, with a wheaten body and salmon breast. Each individual feather appears to have a detailed design. The partridge Wyandotte and partridge Plymouth Rock exhibit this complex and beautiful pattern. Imagine a canvas where the black ink is not only distributed, but creates a detailed design and a drawn effect on each brushstroke, individual feather. The Asiatic partridge palette. This is similar to the wild partridge palette, but with a more pronounced extension of eumelanin, which gives it a generally darker appearance. The pattern of drawing on the feathers is still present, but with more black. Some lines of light Brahma, although the color pattern is modified by other additional genes that give it its unique characteristics, may have this base. Now we're going to talk about the importance of crossing within the same basic color palette. Understanding these palettes is fundamental for several practical reasons in the breeding we want to achieve. If you cross two chickens that genetically share the same basic color palette, for example, two chickens with a well-established solid black base, it's certain that their offspring will also emerge with that same pattern and color base. This is because the genes for the distribution of eumelanin and pheomelanin interact in the same way between both parents, generating consistent and predictable results. Let's look at the following example. If you have an exhibition line Rhode Island Red rooster, known for its dark uniform mahogany, and you cross it with an exhibition line Rhode Island Red hen, the offspring should, by necessity, come out with the same intense color as their parents. This is because both birds have been selected for generations to fix the same wheaten palette genes that intensify and distribute the mahogany color. When you cross chickens from different basic color palettes, the genes that control the distribution of eumelanin and pheomelanin are different in each parent. When combined, the genetic interaction is different, and the result will be a mix or pattern that can be unpredictable, inconsistent, or that does not conform to any recognized breed standard. This is what we refer to as dirty or messy plumage in genetic terms. But if you cross an exhibition line Rhode Island Red, dark mahogany, with an egg production line Rhode Island Red, which is usually a lighter mahogany or red color, the pigmentation genes interact differently. Although both are Rhode Island Red, the production line has not been selected with the same rigor for color. It may have other alleles that dilute or restrict the red, or that introduce more black or white. The result will be a mix of both differences. The chicks could be lighter, darker, or have less uniform patterns than those from the exhibition line. Imagine you cross a White Leghorn rooster, which comes from a palette where genes inhibit almost all pigmentation, with a partridge Wyandotte hen, which has the complex wild partridge palette with an intricate design on each feather. The offspring will be neither a pure White Leghorn nor a clean partridge Wyandotte. It's probable that you'll get chickens with dirty white plumage, gray with irregular black or brown spots, or with messy patterns that don't resemble any of the parent breeds. The pigment inhibition from the Leghorn interacts confusingly with the complex pattern distribution of the Wyandotte, resulting in plumage with no clear definition. Understanding these palettes gives you a solid foundation for predicting the colors of your chicks, selecting the right parents for your breeding goals, and most importantly, comprehending why some crosses produce consistent results and others do not. Now let's move on to the last point for today: the additional color genes, modifiers, and designers. If the basic color palettes we saw in the previous point are the canvas and background color, then there are other genes that act as special brushes, diluents, or pattern designers. These genes don't change the basic palette, but they modify it in surprising ways, creating the enormous diversity of colors we see in chickens. Let's define a term we'll use a lot in this course, and that is locus. What is a locus? In genetics, a locus is the specific and fixed position of a gene on a chromosome. Think of a chromosome as a map, and each locus has a unique address where a particular gene is located. Different versions of that gene, called alleles, can occupy that same locus, and those versions are what determine the variations in characteristics, such as the color of the plumage. We're going to focus on some of the most interesting and visual designers, including the two main ways to get white color. Locus 1. Pigment inhibitor. The off switch for color. Dominant white. This gene is like a master switch that can turn off or suppress pigment production in feathers, even if the chicken genetically should be colored. It's a dominant gene, hence dominant white. If a chicken inherits at least one copy of this dominant gene, its plumage will be predominantly white, regardless of what underlying color genes it has. It's as if the eye switch overrides all other colors. If it inherits two copies of the inhibitor gene, the effect is even stronger, and can sometimes result in complete whiteness, or even be associated with vision problems. A classic example is the White Leghorn. This breed is the best example of the action of the eye gene. Although underneath its white plumage, a White Leghorn might have genes to be red, black, or partridge, the dominant eye gene makes it completely white. What can we get if we make the following cross? If you cross a Rhode Island Red rooster, colored, not inhibited, with a White Leghorn hen, dominant white, the result for the F1 first-generation chicks is that they will inherit at least one copy of the eye gene from the Leghorn. Therefore, they will be predominantly white. However, because they also inherit the color genes, red or mahogany, from the Rhode Island Red, and the dominant eye gene is not always 100% effective in a single copy, it's very common for these chicks to hatch white with small spots, specks, or splashes of red or gold color from the Rhode Island Red, especially on the head or back. As they grow, they may develop contour feathers with some pigment. This cross is not reliably sexable by color at hatching, as both F1 males and females will inherit the eye gene and show that predominantly white coloration with red specks. It's like having a color printer, but someone presses the Print in Black and White Only button, no matter what color file you send it. Now let's learn about Locus B, barred. This expression is known as the zebra pattern and sexing at hatching. This gene creates a very distinctive pattern of alternating bands of pigment, generally black, and unpigmented bands, white, along each feather. It's like a zebra or striped effect on the plumage, giving it a visually striking appearance. If a chicken inherits this gene B, it will express the barred pattern over its basic color palette. The intensity and thickness of the bars may vary slightly depending on the genetic line. It's a sex-linked gene. This is one of its most useful characteristics for breeders. The B gene is located on the Z sex chromosome of chickens. Males are ZZ and females are ZW. This means that the way the barred pattern is inherited can be used to determine the sex of the chicks at hatching by their color, autosexing. Male chicks, ZZ, inherit one copy of the B gene from each parent. They usually have a larger, more defined white spot on the top of their head at hatching, and the black down is lighter. While female chicks, ZW, inherit only one copy of the B gene from their father, because they inherit the W chromosome from their mother, which does not carry the B gene. They usually have a smaller or absent spot on their head, and the black down is darker or more uniform. A classic example is the Plymouth Rock barred. This is the iconic breed for showing the barred gene. Its beautiful black and white striped plumage is the direct result of this gene's action on a dark color base. It's like having a solid colored t-shirt, and then a designer adds precise alternating black and white stripes, and the size of the stripe on the head tells you if it's male or female. We continue with another equally important one. This locus controls the chicken's fundamental ability to produce any type of pigment in its feathers. This locus has two C's, one uppercase and one lowercase. Let's look at their functions. Let's learn about the uppercase C, or full color as it's called. This is the allele that allows the chicken to produce all the pigments, both eumelanin and pheomelanin, that its other color genes instruct it to, like making a color print. Then we have the lowercase circa, which would be recessive white, or white by absence of color. If a chicken inherits two copies of this recessive gene, it will be white. Unlike the dominant inhibiting white, which turns off the color, as in the case of the leghorn hen we mentioned earlier, in this case, the chicken simply lacks the necessary machinery to produce any pigment in its feathers. It's like the paint factory is closed. No matter how red or black it should be genetically, without the ability to produce pigment, it will be white. Classic examples are the Cornish white, the Rhode Island white, and the Plymouth Rock white. These breeds are excellent examples of white chickens due to the presence of this recessive gene. Now let's look at another very important gene, the Locus S, silver or gold. This gene is super important for color sexing, which is key for producing sexable hybrid layers. This gene is responsible for deciding whether the red or gold pigment, pheomelanin, is expressed as gold or red, recessive allele, or as silver or white, dominant allele. It's a switch for the chicken's metallic color type. It's a sex-linked gene and is located on the Z chromosome. This location is fundamental because it allows us, with appropriate crosses, to determine the sex of the chicks by their color at hatching. Remember that in birds, males are ZZ and females are ZW. The S locus is expressed on the Z chromosome and defines sex by color in a specific cross. For males and females to have a different color at hatching, a rooster of one variant, gold, must be crossed with a hen of the other variant, silver. The parental rooster must be homozygous for gold. For example, a Rhode Island Red rooster line that breeds true for gold or red color. The parental hen must be silver. This means that her single Z chromosome must carry the silver gene. What happens with the chicks? Male chicks, ZZ from this cross, inherit a non-dominant Z chromosome from the father, gold rooster, and a dominant Z chromosome from the mother, silver hen. Since silver, S uppercase, is dominant over gold, S lowercase, all male chicks will hatch silver or very light white with silvery tones. Now let's see how the female would hatch. Female from the cross, ZW. They inherit a recessive Z chromosome from the father, gold rooster, and the W chromosome from the mother, which does not carry the S gene. Since they only have the gold gene, recessive S, on their Z chromosome from the father, all female chicks will hatch red or gold. Breeds like the Rhode Island White and the Plymouth Rock White are predominantly white due to recessive white, C, at Locus C. Remember, they don't produce pigment. However, this doesn't mean they can't have other color genes hidden in their genome, such as the silver S gene at Locus S. It's possible that these breeds, if they have been adequately genetically selected, carry the silver S gene on their Z chromosome, despite being white due to the, quote, C-dominant gene. Although the CC gene makes them white, the S gene is still present in their DNA and can be transmitted. Large genetic companies, such as those that develop the Issa Brown, invest decades in extremely rigorous genetic selection programs. Their goal is to create pure parental lines that, when crossed, produce predictable and reliably color-sexable offspring. They use a gold male line that is consistently red or gold, like purified Rhode Island Red or New Hampshire lines, and, crucially, that does not carry the dominant inhibitor I gene. They develop a silver female line. These hens are usually white, but their whiteness is achieved in a controlled and specific way for this purpose. They are either recessive white, C, and at the same time carry the dominant silver S gene, or they are directly silver S, where the dominant inhibitor I gene has been rigorously eliminated from their genome. The emphasis of these companies is to ensure that the I gene, dominant white, is not present in the parental lines, especially in the silver hen line. If the I were present, it would inhibit the expression of silver and gold in the chicks, and color sexing would not be possible. Thus, by crossing the gold rooster with the silver hen, but without the I gene, it's guaranteed that male chicks will be silver or white, and females will be gold or red, allowing for highly accurate color sexing at hatching. We'll briefly mention some other important modifying genes. Any questions about these genes I've mentioned below, you can leave them in the comments. Locus BL Blue, the soft diluter. This gene dilutes black color, eumelanin, to a gray or blue shade. If the bird has two copies, BL, the color is diluted so much that it appears splashed with white. An example of this case would be the blue Andalusian breed. Locus Lav, lavender or mauve, the total diluter. It dilutes both black and red or gold to a pale and uniform lavender or mauve tone. It is a recessive gene. An example would be the lavender Orpington variety. Locus Mo, mottled, the spot sprinkler. It produces distinctive white spots on the tips of the feathers, giving a mottled effect. An example is the Ancona chicken breed. And finally we have Locus ML, melanotic, the darkener. It tends to extend black pigment, eumelanin, making the feathers darker than they would otherwise be. An example is the Sumatra breed. And with that, we've reached the end of our first chapter of this Basic Poultry Genetics course. Today we've unveiled the mysteries of the two basic pigments, how our chickens' color palettes are formed, and the fundamental role of genes like the inhibitor, BARD, color capacity, and silver and gold in their plumage design, and most surprisingly, in sexing chicks at hatching. I sincerely hope this information has opened your eyes to the incredible complexity and beauty of poultry genetics. This knowledge is not only fascinating, but also extremely valuable for any breeder, from hobbyists to those looking to optimize their egg production. Understanding how these genes combine will save you time, money, and allow you to achieve your breeding goals more efficiently without having to pay a fortune for courses that offer less. Remember, we are a passionate community, and this course is for you. Your support is our driving force. If this chapter has been helpful to you, if you've learned something new, or simply enjoy our content, please subscribe to the channel and activate the notification bell so you don't miss the next chapter or any of our updates. Also, share this video with other breeders, friends, or anyone who loves chickens. Let's help this knowledge reach more people. And most importantly, leave all your questions, doubts, or comments below. We want to build this course together and ensure that every concept is perfectly clear. In the next chapter, we will delve even deeper into how these genes interact and perhaps venture into the fascinating world of patterns and mutations. Don't miss it. To your success, fellow breeder. Until next time.